Welcome, everyone. If you don't know already, my name is Robert Driver. I'm a postdoc at Duke University, and welcome to the AOS Student Research Grant Seminar Series here. So each year, the AOS offers funding to students and early career researchers with the AOS Student Research Grant, and we're coming up on the due date next month. And the AOS Student Research Grant Seminar Series celebrates the challenging and innovative work of the AOS Student Research Grant recipients by sharing this work with the AOS members. And each week, we're showcasing the research and research plans of a 2024, 2023, or 2022 AOS Student Research Grant winner in the seminar-style format. And as you may already know, we have one seminar each week for 15 weeks, and this is the 14th week. So one more week after this. And I want to thank everyone for being here. Thank the AOS and the AOS Student Affairs Committee for support and input, and especially thank Alexander, our speaker today. So before we turn our attention to Alexander's research, I want to mention if you have questions, there will be time for Q&A at the end of the talk. And you can participate by raising your hand, the raising hand icon on Zoom, and that will allow me to unmute you so you can ask your question aloud. And you can also ask your question by posting in the chat box or in the Q&A box, and you can do that anytime during the hour, and then we will read those questions out at the end. And I want to remind everyone that this is an AOS sponsored event and that all participants are required to follow the AOS virtual meeting event code of professional conduct. And I'll post a link to that code of conduct in the chat. And I'll also post a link to our post-seminar evaluation form. This allows us for you to provide feedback to the seminar series so we can continue to improve it as we move towards the future here. And also posting a contact email for the AOS Student Affairs Committee. And please be aware that we are recording this talk. Okay, so this week, Allison is currently a Master's of Science graduate and a member of the Macaw Recovery Network of Costa Rica. And Alex's academic career began with a Bachelor's of Science degree in biology from University of Massachusetts Amherst. And during undergraduate, Alex researched vocalizations in chestnut-sided warblers, as well as an honors thesis in vocal amplitude across social context in house wrens that was in the lab of Dr. Jeffrey Podos. And in 2022, Alexander began graduate school at New Mexico State University in the lab of Dr. Timothy Wright, researching vocal variation and development and post-fledgling movements and how they could assist in the conservation of great green macaws in Costa Rica. And that's what we'll hear about today. So Alexander's research in conservation behavior is published in Conservation Science and Practice. And so with that, I'll turn it over to Alexander for his talk, Conserving the Critically Endangered Great Green Macaw from First Word to First Flight, a Study of Geographic Vocal Variation, Vocal Development, and Post-Fledgling Movements.
Okay, thanks very much, Robert, and thanks everybody for being here today. I'm excited to share this work with you. As Robert mentioned, this is my master's thesis work from the last few years at New Mexico State with Dr. Tim Wright, and just want to thank the AOS for helping fund this work and for having me here today for this seminar series. Also want to acknowledge before we get started here, my committee members, Karen Mabry, Martha Desmond, and Jean Fair, as well as awesome collaborators, Macaw Recovery Network and our Manzanillo down in Costa Rica. So as ornithologists, we are well aware of the amazing variety of form of birds, as well as the beautiful plumages. But today, out of that variety, I'm going to be focusing on just one order, the parrots, Psittaciformes. And you may be wondering, well, why parrots of all this diversity? Well, they are charismatic flagship species, oftentimes for conservation efforts. They're incredible vocalists. They're highly social. They have important ecosystem services, such as being seed dispersers and pollinators. And unfortunately, because of the fact that they are such incredible vocalists and have beautiful plumage often, they're one of the most threatened orders of birds. So staying on this theme here of one of the most threatened orders of birds, that comes from two main sources, habitat loss and nest poaching. And indeed, the IUCN estimates that 38% of parrot species globally are listed at some level of risk from near threatened all the way up to critically endangered. So what can we do about these population declines? Many species are in decline. What are some conservation strategies we can implement? Well, we can protect and restore habitats. That's one of the things we can do. We can engage communities. We can build community programs that raise awareness about these birds that may be in decline and help with initiatives on the community scale. We can also create captive breeding programs for species that may need eventual reintroductions of birds into the wild. So on the theme of habitat protection here, what might we need to do? Well, we could identify areas used during the annual cycle of a species, say breeding, non-breeding, or migratory habitat, assess the characteristics of those areas, and relate those characteristics to then what we know the species requires. And if there's a discrepancy there, we can begin restoration activities and eventually, hopefully, protect areas from further degradation through either federal designation or private purchase of lands. And for reintroductions, what might we do there? Well, they are usually a last resort because, of course, we want to do as much conservation work as we can in situ, but unfortunately, they are becoming increasingly necessary. And many factors must be considered before reintroduction can take place, such as selecting the individuals who will be released, performing a habitat assessment, and pre-release trainings, making sure that the birds know which predators to avoid and which foods to forage on, as well as post-release supplemental feeding and monitoring activities to ensure that birds are integrating into the wild. So what questions may arise from habitat protection and reintroduction type studies? Well, for habitat protection, we may ask, what habitats are individuals using during their annual cycle? We do want to know where they are so we can make sure to effectively protect habitats that they're often using. And then we may also ask, where are the knowledge gaps in their movement patterns? And for reintroductions, it's important to know, are they even appropriate? Is this a strategy we want to pursue? And then what information is needed to increase their success? So the species I'll be focusing on today is the Great Green Macaw, or ambiguus, and we'll be exploring some of these questions about movement and vocal behavior in the context of reintroductions and habitat protection. Now, this species ranges in 6 countries from Honduras down to Ecuador in 2 different subspecies, the nominate ambiguus in the north and the much smaller population of Guayaquilensis in Ecuador only. And last assessed by the IUCN in 2020, estimated that there were between 500 and 1,000 mature individuals in the wild, and officially listing them as critically endangered. Now, over the past few decades, lots of work has been done on the species. Research has focused on diet, nesting habitat, migratory movement, breeding behavior, taxonomy, genetics, phylogeny, and the development of multinational conservation efforts among countries within the range. But 2 areas that have either not received focus or haven't received focus in recent years are vocal behavior and post-fledging movements. And these are the 2 themes that I'll spend the rest of my talk focused on today. And we'll start with vocal behavior. This was the first chapter of my thesis entitled Vocal Development and Patterns of Vocal Variation, Informed Conservation of a Critically Endangered Macaw. So as you may be well aware, parrots are amazing vocal learners. Specifically, they have remarkable imitative vocal learning abilities. And they're also open-ended learners, meaning that unlike songbirds, they have the capacity to learn vocalizations throughout their lives and not only in an early life sensory learning period. And they use these vocalizations for a number of important functions, such as coordinating foraging and roosting movements, defending nest sites, and facilitating reproduction and pair bonding activities. Vocal development in parrots is also an area that hasn't received much research historically in the wild, though more studies are occurring in recent years. And there are fascinating parallels in vocal development in parrots between parrots and humans. Namely, early in development, research has suggested that innate vocal production circuits lead to few call types with little variation, and begging calls tend to predominate. Whereas in mid-development, vocal learning through exposure begins to become more apparent. And we see the overproduction of call types. And there may even be babbling that occurs in some species where auditory feedback is implemented to solidify that vocal repertoire. And then later in development, we may see selective attrition of the repertoire, where the repertoire then shrinks and contact calls are refined for later use in social situations. A phenomenon that can develop in vocal learners is that of vocal variation, whereby during learning, receivers may make copying errors that then eventually leads to vocal variation across generations, where you may see different vocal types across the landscape within a species. And geographic isolation, whether it be human-caused or not, can accelerate vocal differentiation, and you may begin to see acoustic differences among populations. And this has been well documented across many parrot species, from the yellow-naped Amazon to the Gala to the orange-fronted parakeet, just to name a few here. And vocal variation may have an impact on reintroductions. Captive breeding populations of many species that are being bred for reintroduction purposes are often isolated from their wild counterparts. And they may even include human-raised birds, and often do, that have varying social histories. Vocal divergence has already been found to occur among different captive and wild populations of one species of parrot, the Puerto Rican parrot. And we also have seen in a wild context that translocated yellow-naped Amazons that have been moved from one dialect area to another sometimes self-segregate within their dialect type. And these bodies of research suggest that vocal variation between introduced and wild birds may be an important factor to consider during reintroductions and at release sites, but it is seldom considered today. And overall, communication barriers between two groups of birds could prevent the information sharing really necessary for introduced birds to successfully integrate into the wild population. So today, what are we going to focus on? Well, we want to know, do adult great green macaws show variation in contact call acoustic structure among different geographically isolated Costa Rican populations? So here are three hypothetical populations here. We're curious, do they all sound similar, or do their calls bear a population-specific signature? And then from the vocal development side, we're interested in, among those same isolated populations, how are nestling calls developing, and is vocal variation present in those calls? So how are these calls developing, and do those nestlings themselves, as they're developing in the nest, bear that population-specific signature? And for the rest of the talk, I'll use this icon in the upper right corner of the slide when I'm talking about the adult part, and this icon for nestlings. Okay, so here's a map of our study area down in Costa Rica. In the north, we have the wild population that we looked at outlined there in that polygon. And then we also worked on the Caribbean coast at a reintroduced population at a facility run by our Manzanillo, and then over on the Pacific coast at a captive breeding center run by the Macaw Recovery Network. And interestingly, birds at both the captive and reintroduced centers were originally from the same source population, and they were split to their respective coasts in 2010. And prior to that split, it's unknown how many of those birds may have come from this wild population. So for adults, then, we may predict that because of this obvious geographic isolation, variation may exist among these birds. Alternatively, it may not exist because these centers have birds from that shared source. And for nestlings, we may predict that vocalizations will develop along similar trajectories, regardless of the social environment and what population they may be in, and that, unlike adults, variation will be less detectable during that period in the nest when vocalizations are forming. So for adults, we recorded them in all three populations from February 17th to April 13th, 2024. You can see here on the left there, me recording in the wild population, and at the right at the captive breeding center. For analysis, we visually selected contact calls using RavenPro software and analyzed each of these selections using the Warbler package and R, bandpass filtering and tailoring each call to exclude low and high frequencies that may interfere with the analysis, and then extracting 26 spectrographic parameters from each call, time and frequency measurements, and then running spectrographic cross-correlation on the entire data set, which is a technique that slides spectrograms on top of each other to get a correlation coefficient of how similar they are. Then we conducted principal components analysis on those spectrographic parameters and ran multidimensional scaling on the SPCC results to describe how similar calls were across populations. And finally, tested for population differences statistically using a combination of linear mixed effects models and Mantell tests. We recorded nestlings in those same three populations from February 21st, 2023 to May 18th, 2024, using a combination of different models of autonomous recording units, or ARUs. And these are pretty cool little devices. If you're not familiar, you can program them to record during certain times of day, and they store the information on board. Here's what one looked like mounted inside a wild nest cavity, outside a nest box at the captive breeding center, and underneath an artificial nest box in the reintroduced population. We then aged nestlings based on a combination of nest monitoring and morphological characteristics. For analysis, we again selected these calls visually using Raven Pro software, sampling calls every 4 days to track development patterns in these nestlings. And then we followed the same two acoustic measurement pathways that I described for adults. Finally, testing for both age differences and population differences using the same two types of tests. Okay, so we'll get into some results here then for the adults. Overall, we recorded 104 hour-long tracks of adults from the three populations. And after review of about 10.5 hours of good quality data, we were able to select 289 calls for analysis. And you can see here the breakdown by population. Here's what some of those adult calls looked like. So we have 3 calls from the wild population on the top, captive in the middle, and reintroduced on the bottom. And each call is standardized to the same time and frequency measurements, with time in seconds on the x-axis and frequency in kilohertz on the y-axis. And you can see that these calls consist of harmonic stacks, where most of the energy of the call is concentrated at pretty low frequencies. And those 2 dashed red lines in that upper left call are the bandpass filter limits that we used for analysis. Now, just to give you a sense of what these calls sounded like in the wild, and to show a brief video of a group of about 15 adult great greens flying in the north of Costa Rica, you'll be able to hear in what context they use these calls. So pretty cool to be able to witness a group of them using that in the wild to coordinate those social movements. Okay, so then when we turn to looking at the results from our principal components analysis, this is a technique that summarizes all the variation that we may have captured by measuring 26 different spectrographic parameters from each call and reducing it down to dimensions that we can both test statistically and view in 2 dimensions, as we're about to do here. And you'll see a bunch of dots will pop up. Each dot will be a call, color-coded by population there at the bottom. So what we see here is pretty high overlap among calls from all 3 populations. You can see that there's more variation from the wild population and the captive population as opposed to the reintroduced population, but we do see pretty high overlap among calls from all 3 populations in this acoustic space. And indeed, when we run our linear mixed effects models, they do not find significant differences between these populations for either of these PCs. And when we look at our other analysis technique, multidimensional scaling, this is the last step in converting those correlation values from the SPCC data down to points, again, in a 2-dimensional coordinate system that we can view and test statistically. And again, here you see pretty high overlap among calls from different populations. Here we're demonstrating overlap with contour lines, where lines closer together indicate more calls in that area of acoustic space. And when we perform our Mantell test, which looks at correlations between the population identity of where the call came from and its acoustic similarity to other calls, we see a pretty low correlation coefficient, again suggesting that there is high similarity of calls from across these populations. Okay, now looking at our nestling results here. We recorded about 7,500 hour-long tracks of nestling calls from the 3 populations. The beauty and downfall of ARUs is there's a lot of data to process. And we reviewed a lot of that data and got 460 hours or so of good quality calls, selecting 2,927 for analysis. And you can see again here the breakdown by population and the number of nests we were able to include from each population. And in all, we saw a similar vocal development trajectory supporting our first prediction across populations. This is an example of a lexicon from one wild nest, where you can see week on the top, starting from less than or equal to 2 weeks, all the way up to 9 to 10 weeks on the right. And for context, these chicks are fledging at about 12 weeks of age. Again, we have frequency on the y-axis, time on the x-axis. And I'd like to just play now a few of these so you can get a sense of how they change acoustically over the course of development, which I think is pretty cool. So calls from that youngest age group, less than or equal to 2 weeks, tended to sound like this. From 3 to 4 weeks, we see a pretty significant shift. And then again from 7 to 8 weeks. And here you see these calls are starting to look much more like the adult contact calls. And then finally at 9 to 10 weeks, we get these wide broadband grunts that start to appear. So now we'll look at the principal components analysis, same technique as we showed for the adults, for nestling calls by age. So rather than by population color-coded now, we're going to be looking at all these nestling calls pooled within these age groups to get a sense of how these are changing over the course of development. So I'd like to draw your attention, there are a lot of points here, to two areas. One is this clump of points here of calls from the less than or equal to 2 weeks age group. And the other is a cluster of points here from the 9 to 10 weeks age group. And when we look at this by each principal component, we see that for our first principal component, which is summarizing the most variation in the data, 5 frequency measurements were most important for describing that variation. And indeed, when we run our linear mixed effects models and our post hoc tests, we see that calls from that less than or equal to 2 weeks age group and 9 to 10 weeks age group that we'd circled before were significantly different in those most of those comparisons to other age groups. For our second principal component, we found that 5 timing measurements were most important in explaining variation in the data. However, there was more variation in whether pairwise comparisons between age groups were significant or not. When we look at our other analysis technique, multidimensional scaling, we see some interesting things here. First, we see that calls produced early in development in this blue, less than or equal to 2 weeks age group, and a few calls in that 3 to 4 weeks age group in yellow there, are highly different from most other calls. And then as nestlings continue to develop later on, calls produced differ less. And additionally, variation seems to decrease as fledging nears, as indicated by the tightening of those contour lines in older age groups, suggesting that as fledglings are approaching, or as nestlings are approaching fledging rather, that variation is decreasing in their vocal repertoire. And when we run our Mantel test here, we find a pretty high correlation of 0.58 between the age group and the call, suggesting that there really is clear separation here. And then when we look at the same multidimensional scaling technique by population, this is getting at if we can see vocal variation across populations at the nestling stage. So here we've broken it down by age group, and we'll look at each of these within our population framework, and I'll show some Mantel test results that we'll focus on. So starting with this less than or equal to 2 weeks age group, we see a pretty low Mantel test correlation coefficient of 0.2, suggesting low similarity. And then that really increases to 0.64 at 3 to 4 weeks, and then decreasing again to 0.17 when those chicks are getting a bit older from 5 to 6 weeks. And I'll go into discussing that in a few slides here. So in summary, we saw minimal vocal variation among adult populations of adults, where our PCA and MDS analyses showed high visual overlap among populations. Our mixed models did not find significant differences, and our Mantel test found only a weak correlation between structure of the call and population that it came from. This and all suggests minimal variation in contact call acoustic structure among populations. Certainly no evidence of discrete dialects has been described for many other parrot species, and that it would be unlikely that communication barriers would form if birds from these populations were to come into contact with each other. So why do we not detect vocal variation? Well, it could be quite simply that the species doesn't develop it among populations. This has been noted in other parrot species, such as the monk parakeet and the thick-billed parrot. It could also be that vocal variation does exist, but we just did not sample widely enough in geographic space. And a future study could look at this same question across the species' range. And the third explanation could be, and this is the one that I think is most likely, is that this minimal variation could be due to the limited time in isolation that these populations have been in, only about 14 years. We also saw that there is vocal variation in nestlings, but it varies by age, where we saw a greater difference across populations in that 3 to 4 weeks age group, and smaller differences across populations in those two age groups on either side. What might explain this? Well, in those youngest birds, it could be that the production of common innate calls across populations suppresses vocal variation from forming. Whereas as nestlings tend to age a bit more, and we see that greater difference, it could be that the overproduction and potential babbling leads to increased variation in learned calls in their repertoire. And finally, as chicks are aging even more and we see a smaller difference, it could be that calls are beginning to converge because of both the local matching pressures that these birds may be facing as they leave the nest and selective attrition of the vocal repertoire. And a finding that I think was pretty cool is that we saw that great green macaw nestlings seem to babble. And this is the production of vocalizations outside of a clear behavioral context. You can imagine this as an infant or toddler human making these sounds, right? And it's pretty cool because this phase sits at the junction between the innate calls and learned calls in these birds' repertoires. And the auditory feedback from this babbling, as has been demonstrated for many other species, may be critical for fine-tuning that vocal repertoire. And I think this is an important finding because we see this phenomenon across other parrots, such as the green-rumped parrotlet and the kea, passerines such as the zebrafinch, other mammals such as the humpback whale, and of course in humans. So what might we want to do for future research for the adults here? Well, dialects, we know in other critically endangered species, the yellow-naped Amazon, are shifting in response to declining populations. And because population connectivity is a main driver of vocal convergence in a lot of these species, it could be that vocal variation will develop among increasingly fragmented great green macaw populations in the future. So future studies could record vocalizations throughout the range of this species to evaluate the feasibility of broader reintroductions. And I think it'd be important to conduct a follow-up comparison among these same three populations in the next 5 to 10 years to see if time is what's preventing vocal variation from forming at the moment. However, at this point, we suggest that reintroductions can continue without risk of communication barriers forming. And for nestlings, we saw that their calls are developing along similar trajectories across populations, indicating stable developmental stages regardless of social environment that these birds are in. Though future work could audio and video record these vocalizations from all age groups to fill in some of the gaps we had in our analyses, as well as, I think importantly, assigning behaviors to call types. We also saw that vocal variation is most apparent during the overproduction stage of vocal development. And I think this emphasizes that investigating vocal development across different populations of parrots, whether they be isolated wild birds or captive and wild and reintroduced as we had here, can help us understand what patterns or lack thereof variation we see in adult birds. Okay, so I've just spent some time talking about vocal behavior in the species, and for the remainder of the talk, I'll turn to post-fledging movements. And we'll also discuss, as this title suggests, some limitations of current tracking technologies for these large parrots. So if you're a recently fledged bird, this is a very vulnerable time, right? The environment is completely novel. You have high mortality and there's stress from unpredictable climate conditions and predators. And it's also a very interesting time to study because the dependence period for fledglings on their parents varies in length widely across bird species. This is the period where they depend on their parents for food, and movements are also very limited. So the habitat into which these birds are fledging is both important to understand and protect. Historically, most post-fledging studies have focused on passerines and raptors, with fewer studies focusing on post-fledging and parrots. And this period in parrots is really critical to understand because of the many different threats that parrots are facing, and the fact that many of these declining species urgently need juvenile recruitment for their wild populations to sustain themselves. And historically, descriptions of post-fledging movement patterns, sources of mortality, and social group dynamics are lacking for many parrot species, and particularly for some large macaws. And understanding which habitats these fledglings are using, as I mentioned, can help target conservation efforts. So VHF radiotelemetry has been the norm for past parrot movement studies. However, required labor is quite high for this technique, and the ranging movements of many of these widely foraging birds inhibit detection probability. However, remote transmitting GPS units are becoming available, which is exciting. However, they're often quite expensive, sometimes 10 times as much as a one VHF radio collar, and more susceptible to destruction, particularly by these large parrots with their strong beaks. And in the field, these techniques also look quite different, right? So you may have a set of VHF radio collars out, but you may be spending most of your time running around looking for where these birds are, right? Only to say, where are they? However, with these more expensive modern units that are becoming available, you may be able to just sit at your desk, right, with a cup of coffee and have these data beamed directly to your desk, right? And often in very large, high resolution quantities. And though it may seem like the remote transmitting GPS unit is the easy choice here, radio collars do remain the feasible option for many studies because of their much lower initial investment and the fact that they're often more durable for large parrots. In the past, there's been work on great green macaw movements using radio telemetry conducted by Centro Científico Tropical in northern Costa Rica from the mid-90s to the mid-2000s. And they did a lot of important work establishing breeding areas, migratory movements, and non-breeding areas, including working to establish a corridor of protected areas now known as the San Juan La Selva Biological Corridor. However, the landscape and climate in that area and throughout the range of the great green macaw continues to change. And the current knowledge of this vulnerable period of post-fledging is important to understand to make sure that we're continuing to protect areas that these birds are using. And now the Macaw Recovery Network has been monitoring the breeding population there in Costa Rica for a number of years. So we teamed up with them for this tracking project to ask a couple questions. The first being, what movement patterns are great green macaw fledglings exhibiting in northern Costa Rica? And then doing a bit of a dive into what telemetry units are currently available for large macaws. So again, this is the same map of the wild population I showed for the vocal work there in the north of Costa Rica. And we begin by nest monitoring, and MRN has been doing this for a number of years in that area, monitoring nests and noting which ones are productive each year. And if breeding activity was noted, we would ascend to each nest every 1 to 2 weeks to check the number of chicks and continue to estimate their ages. And we would use a rope ascension system, as you see here on the right, to get to those nests. If there were chicks in the nest, we would age them based on a combination of body mass and feather growth. And once they were at least 8 weeks out of that 12-week nestling development period, we would fit them with a VHF radio collar that included a mortality beacon so we could hopefully estimate if they reach mortality once they'd fledged. We also, through a generous donation, received some low-tech satellite transmitters that we mounted on some radio collars. And the overall radio collar satellite unit and mount weighed approximately 45 grams, or no more than 4% of chick body weight. And we made sure, of course, that this fitting was loose enough to both account for remaining growth the chick would experience in the nest, while tight enough to not slip over the head or become caught on vegetation once they'd fledged. Here's a picture of a probably 8 or 9 week old great green macaw nestling with that radio collar on, already beautiful and quite large, as you can see by my friend's hands there holding it. And here's what that satellite transmitter looked like, fitted on top of that radio collar. And you can see there a quite long antenna running down the back of the bird, and that's necessary to transmit location information to satellites. So to conduct manual telemetry, here's a photo of me doing that. we would first confirm whether birds had fledged, and we conducted telemetry from the end of the breeding season all the way for a few months, and then on a return trip later in 2023, sampling first near nesting areas, and then later from high elevation viewpoints, both to maximize detection probability with the radio telemetry system and account for previous movement studies suggesting that these birds are moving to higher elevations post-fledging. And what did our movement data look like then? Well, we may have negative detections. Indeed, we had many of these where we would scan for an individual and come up with no signal. We may have single bearing detections where we would receive signal for an individual from one direction, but not be able to effectively triangulate. And then sometimes we would be able to triangulate where we would have at least two or more locations of an individual that we'd then be able to calculate its approximate location. And then, of course, the best of all, a visual location where we'd be able to track a bird to a place where we would see it and identify it with a radio collar on. So how did we do? Well, we monitored 31 potential nests. 22 of those ended up being occupied. And these nests primarily occurred in the Great Green Macaw's favorite tree, the mountain almond. And they used this tree both for nesting and as a large portion of their food sources. And interestingly, most of these nests occurred in areas like this, where they were surrounded by pasture, perhaps with scattered isolated trees in the area. Of the 22 occupied nests, we selected 18 for our study, and we fitted 24 chicks from those 18 nests with VHF radio collars. And then we had those 6 of 24 also fitted with satellite transmitters. And of course, we're all bird people, right? We love looking at photos of cute birds, and here they are. These are the Great Green Macaw nestlings that we fitted with radio collars. It was a really great time being out there in the field with these guys. Okay, now we'll look at some maps here, and you'll note that the backgrounds of these maps have been removed, and this is to prevent potential poaching risk, which is a real issue for this species. So what we're looking at here is an overall summary of where we went to attempt to detect these birds. So each brown triangle is a nest, and each blue dot is where we went to scan for the birds that we tagged. And you can see there, Indio Maíz Biological Reserve and Tortuguero National Park, those are two locations where Great Green Macaws also occur in nearby areas. So out of 418 detection attempt sessions, that's the number of times we went out and we scanned for all birds. We scanned almost 2,000 times for the birds that we tagged. And as you can see, quite a low number of those were positive detections, or only 4.8%. Here's that same map of nests in the brown triangles, and these are our successful detections. So you can see many fewer points, and these are areas, these are locations where we estimated that birds were in the blue dots and visually located them in the green dots. And in all, we triangulated 13 of our 24 fledglings, a total of 16 times, and visually sighted 4 of them 7 times. We detected mortality signals for 4 of those fledglings. One of those ended up being a dropped collar in the nest, which we recovered, and the other 3 we triangulated to be within 1 kilometer of the nest, but because of either the terrain or access, we weren't able to recover those collars, so we're not certain about the fate of those birds. And perhaps one of the biggest bummers of this part of the study is that we did not receive any data from our satellite units despite successful pre-deployment testing of these units. And I really think that's because of the long antenna down the backs of these birds, the fact that these birds were spending time in the nest pre-fledging, and very easy access for the parents to successfully disable that unit before they'd fledged. Okay, now we're going to look at 3 nests that we did have some positive fledgling data from and some interesting results. Here's a nest in the middle of the screen here, again in that brown triangle. We estimate that the bird from this nest fledged on April 12th. And interestingly, we can see just within a few kilometers of the nest that we located that bird 2 months after that, and then again 3.5 months after that. indicating pretty high nest site fidelity during the 6 months post-fledging for this individual. For this second nest here, you can see again that brown triangle of the nest in the top of the map there. We estimate that these birds fledged around April 21st, and then just within about a month or so of fledging, flew almost 40 kilometers to the highlands there, where we located them on June 2nd. And then, interestingly, almost as far back towards their nest where we located them again just 10 days later. So pretty long distance movements for these young fledglings. Finally, for this nest on the other end of the distance spectrum here, we see that within about 6 weeks of fledging, we located this chick 6 times within just a few hundred meters of the nest. So on a much smaller scale. So interesting differences that we're seeing across family groups. So overall, we saw that fledglings showed pretty high fidelity to their nesting area, though over quite different timescales. And when we did witness birds in the field, we witnessed that they were traveling with their family groups, which is common across many parrot species, particularly larger parrot species. We did also see that some chicks disappeared from their nesting area soon after fledging, while others remained near their nests for weeks. And pretty cool, a whole year later, we witnessed a juvenile with a radio collar on in one of the original nest trees, right, displaying a begging behavior that we've seen from old chicks to their parents, who are now trying to raise a new clutch of eggs. And so birds will come back the next year and attempt to keep getting the parents to help them find food, right, even though they can certainly do it on their own at that point. So detections largely ceased in early June. So we started tracking in late April. We had about 5 or 6 weeks of days in which we were receiving locations. And you can see here from this map, this graph where the points disappear starting in early June, that we had very few detection locations after that. Interestingly as well, based on the size of these dots here, which corresponds to the distance from the nest that we detected birds, the average distance between the nest and the fledgling location increased from 1.17 kilometers in May all the way up to 17.67 kilometers in June or later. So we were detecting birds ranging farther and farther from their nests. And it could be that these positive detections and visual sightings decreasing suddenly was in response to changing food availability. That year, that breeding season, we experienced El Nino weather patterns, which caused a very dry, typically wetter couple of months. And it could have been that the food availability across the landscape shifted because of this, causing birds to range further away from their breeding areas. And the brief returns to the breeding area that we did see for some species may be to either maintain nesting territories or track food resources around the nest, which has been found for other parrot species as well. We also asked the question, as Tom Lewis and colleagues did in their 2023 paper, are these isolated nests an ecological trap, right? So as I mentioned, a lot of these nest trees, one of them shown here, tend to be in these relatively open areas. And fledglings are therefore dispersing into these areas. There could be reduced predation in the nest from non-volant predators, perhaps, that require canopy connectivity to actually reach those nests. Or on the other hand, there could be increased predation from avian predators, right? Much more exposure from the sky. And of course, increased accessibility for poaching if these nests are more able to be found. So it seems like a lot of these macaw parents are selecting for low canopy connectivity, right? But at what cost? And so we recommend that future restoration efforts consider this trade-off between isolated and connected nest trees, because both have their benefits when conducting habitat restoration. So our conservation implications here that we suggest are that the area around the nest seems to remain critical even weeks after fledging. We saw birds in the area sometimes months after fledging. And interestingly as well, from field observations, we saw that parents seem to choose consistent locations to leave fledglings when they go off to forage, and they're often large isolated trees. Why might this be? Well, they could be notable landmarks, right? The parents could be using these trees because they're easy to find again after they finish foraging. Or it could be because they're mimicking the characteristics of nest trees, which are often also large and isolated. And again, it could be because they're protecting from non-volant predators that may rely on that canopy connectivity. So therefore, we recommend preventing the felling of these so-called daycare trees that remain around the nest. It may be easy to assume that if you leave the nest tree and fell all the trees around it, that it will be okay. But indeed, it seems like these isolated trees in the vicinity are important for these macaw families. And just want to return to finding remote transmitting units for macaws. This is an extremely difficult task if you've ever worked with these birds. The detection success we had was quite low, as I discussed, and we didn't even locate 46% of our fledglings. Prior to this study, we did review available telemetry options for all parrot species and macaws more specifically by consulting the literature to see what other researchers had used in the past, and by conducting a widespread internet search of telemetry companies. In that search, we found 15 companies that manufactured 83 different telemetry models, which is quite a few, but only 5 of those companies said that they would sell us units for Great Greens, though many of these often prove to be untrustworthy based on past publications. And the unwilling companies, those 10, always cited the power of the macaw beak. So it was more often than not that I would call up one of these telemetry manufacturers and tell them the species I was working with, and they would decline me right then and there. So we've identified, and many past studies have as well, this great need within the macaw research community for a durable transmitter that's often light enough to fit on these birds, and is remote transmitting that can help us reduce the immense field labor required to capture successful detections. Okay, so just want to move to wrapping up here by going back through and summarizing some of these results as we've discussed. So back to the vocal work, we saw that geographic variation in adult calls is minimal across these 3 populations, suggesting that releases can proceed without risk of barriers forming. We saw that geographic variation of nestling calls fluctuates with nestling age, suggesting that there are vocal matching and selective attrition pressures that these nestlings are facing as they continue to develop in the nest. We saw evidence that nestlings are babbling, which is a very cool finding, and that calls are developing consistently regardless of social context, suggesting that babbling is important for vocal repertoire development in this species. We also saw variable post-fledging movements among family groups that suggested high nest site fidelity and importance of nearby daycare trees. And we also saw a sudden decrease in successful detections, which could be corresponding to a reduction in food availability, causing a widespread departure. And we emphasize that high-resolution spatial data is very important when working with these birds, emphasizing that macaw researchers really need a remote transmitting unit for successful future studies. So returning to this collection of 4 approaches to conserving parrot species, I just want to close by saying that the 2 organizations I worked with, the Macaw Recovery Network and Manzanillo, are engaging on all these fronts down in Costa Rica and other countries in Central America, and are really doing amazing work to conserve the Great Green Macaw down there, as well as other parrot species as well. So just want to close, therefore, by thanking the many friends and family members and collaborators at the Macaw Recovery Network and Manzanillo for their great support with this work. The NMSU Avian Migration Program, which provided funding support for me during the course of my master's, as well as the Mesilla Valley Audubon Society, and of course, most significantly, the American Ornithological Society for funding a large portion of this work and having me here today to present on it with you all. And I'm happy to take any questions, and if you'd rather email me after the fact, my email's there. Thank you.
All right. Thank you so much, Alexander, for that great talk. I hope everyone enjoyed that as much as I did. Really appreciate the clear description of the results and some of those very nice macaw baby images like we're looking at right now. Um, yeah, if anybody has any questions, feel free to raise your hand or use the Q&A box or the chat box. I'll be monitoring all of those. Um, but if not, let me bring the Q&A up. Oh, yes, Tom Lewis, I'm going to allow you to talk. You can go
Can
ahead and unmute, Tom. Yep, great.
you hear me? Brilliant. Yeah, thank
Yes,
you.
we can.
Thanks, Alexander. That was really, really interesting. Both sides of it, both the kind of the vocal side and the conservation side. I mean, there's so many questions I have, but I think I'll just pick one from the kind of the start of the talk about the different vocalizations from the three different populations. That picture that you put up there of the kind of the contact calls, there was obviously there's lots of similarities, and you know the kind of the variables that you looked at were kind of those acoustic kind of things that you could measure. But when you visually look at those calls, I'm not sure if you kind of picked ones that were purposefully kind of a little bit different. You could really see differences in some of the structure. The ones from the captive breeding center, those harmonics looked a little bit more kind of, I don't know, blurred maybe. And the ones from the reintroduction site, there was almost like a little drop, little kink in the end there. And I know from my work when I did stuff on the Great Green Macaws and also the Scarlet Macaws, the Scarlets are more that kind of blurred kind of bit in the middle. I was interested in the captive breeding center is obviously where there are Scarlet Macaws, and you talked about that kind of learning, especially the open-ended stuff. But I wondered, do kind of any thoughts on kind of potential maybe other ways to look at those calls and analyze them, and maybe if those kind of things have influenced those different calls.
Yeah, yeah. Thanks, Tom, for that question. That's a great question. Just reemphasizing what you said for everybody else, at that captive breeding center where we recorded Great Greens, there are also scarlet macaws there. And so certainly potential that the Great Greens could be picking up some of those scarlet macaw call dynamics, which are pretty similar. I think in the future, it would be really cool to get a sense of if the scarlets are in turn taking some of the great green information, call information, to see if that goes both ways. But in terms of our call analysis, the two different ways that we measured calls, both all the acoustic parameters as well as spectrographic cross correlation, combining those analysis techniques made it so that results were overall more robust. And the SPCC is pretty good at discovering anything that the parameters may not have initially picked up on.
Brilliant, cool, thank you.
All right, thanks a lot, Tom. And I was just wondering if any of the nests or if any of your analyses you could pool it by nest. So could have any of the analyses been washed out or results being changed if, say, one nest had a larger clutch size or more measurements? I know there's a variety of different analyses, so more measurements were taken from that nest, which is contributing more to the signal than another nest in the population?
Yeah, that's a great point. So we did have some imbalances like that, and we did control for number of calls from each nest in our models.
Got you. Thank you. Um, and I was just wondering, so the contact call is one type of
Mhm.
call, so that was an initial filtering where all the other sounds produced by the macaws are filtered out then?
Yeah, so the those contact calls of the adults that I showed is the
Mm.
main call in their repertoire. It's the call they give most often, and it's the one that they use to coordinate these movements across the landscape. So because of it being the most prevalent call in their repertoire, and also the one that other researchers have looked at for other parrot species, is that's the reason we chose that one. But yes, we did filter out the other calls that the adults gave. Yeah.
I understand. And then just looking at the questions here, I think this is a very small detail, but in the 5 to 6 week call measures graph, I don't think it's necessary to go back to that slide, but if I'm remembering correctly, the captive population was absent from that graph.
That's
Um,
right.
was that was that a limitation? I was just surprised because I would have thought the captive population would have been the easiest one to obtain the recordings from. So,
Yeah, that's a great point. So yes, the captive population was absent from there because unfortunately we weren't able to successfully measure enough high quality calls during that period. Indeed, actually, in that area, there's a lot of other noise because there are other nests nearby that we had to control for to make sure we weren't selecting calls from those nests, as well as
mm,
many adults in the area that oftentimes would drown out high quality calls. Yeah.
thanks. Thank you. And now I'm going to allow Bruce Byers to talk. So it looks like, yep.
Uh, Great talk, Alexander. It's wonderful to see what you've been up to since your UMass days. I was wondering a little bit about the homogeneity that you found among the adult calls, given that the birds start learning in the nest and then stay in family groups. You'd think this would result in each family having its own, each extended family and its descendants having its own little dialect. What do you think's going on to restore the homogeneity even between the isolated populations?
Yeah, thanks, Bruce. Great to hear from you. That's a great question. So I think what's going on is that in the wild population, where you might expect there to be the most family group level of call variation to occur, these birds are all getting together in the non-breeding season and exchanging vocal types. So they are very much in their family unit during the breeding season, but then return to these very large groups in the non-breeding season. So there's ample time to restore homogeneity to the call pool there. And then in each of these captive and reintroduced populations, birds are, when they're not breeding, they're kept in these very social environments as well in large groups. So there's a lot of opportunity for exchange to occur.
Okay.
But I think that's one of the reasons that sampling across wild populations throughout the range, for example, in populations that have never come into contact with each other would be really interesting. For example, comparing birds from Honduras to birds from Costa Rica, I think we much more expect to see variation there.
Yeah, thanks.
All right. And Tim Wright says, terrific job, Alexander. Thanks for hosting the series. And we appreciate that, Tim, who was Alexander's advisor on this research. So that is great. And I just have one small question related to what you were saying, Alexander, and then I think we'll wrap it up here. So there must be contact calls of this bird on like Xenocanto, or this bird is in the zoo all over. I would guess maybe not as much as scarlet macaw or something like that, but um, can you use those recordings to help with with those questions?
Yeah, that's a great point, and we've thought about doing some of that work. And my advisor, Dr. Tim Wright, has also done some recording of great greens in other places. A future project, if not something I would work on him with, could certainly use those data sets that are out there already. And as long as they're high quality enough, that's a big thing to consider with some of those, Cantor recordings, and as well as if they're available in WAV format as opposed to just an MP3. And so, yeah, that would be a great way to get around some of the difficult field work needed to reach some of these populations.
Yeah, I was imagining how challenging it is to get up to those nests. So, and then you showed the rope structure. So, all right, everyone, we're at the top of the hour here. So thanks a lot, everybody, for being here. Thank you so much, Alexander, for a great presentation on these macaws, and we will see you all here next week for our final seminar of the semester. So bye, everybody.
You wanna-